Hello, welcome! In this AI coffee break, we are going to see how a language model, usually applied on natural language, can solve biology problems. Is this not at least a little bit crazy? So far, in the Transformer series, we have seen how we can use transformer-based language models like BERT or GPT-2, GPT-3 on text, or on images, or even on both. But what if Miss Coffee Bean tells you we can use transformers for analyzing protein structure? So in this video we will explain what the biological problem is and why we can solve it with a transformer model originally meant for language. This method we are talking about was published quite recently. Oh, come on, who are we kidding? It was published 1 month ago. 1 month ago in machine learning is way old. Anyway, let's get back to the subject. What does this paper do? It takes BERT, a transformer model like we presented it in a previous video linked below. We remember BERT is trained on masked word prediction, and one of the most important internal mechanisms is self-attention. Self-attention computes importance scores between tokens, words or subwords in the same sequence. Attention computes importance scores between tokens from different sequences. And we do not compute one attention per sequence, but multiple scores called attention heads, here color-coded. By learning to predict words in natural language, the model learns more complex relationships present in language, like for example parts of speech, coreference, sentiment, and so on. Because BERT is notorious for learning implicit patterns in the data, for discovering structure that is not even annotated, a whole subfield emerged called BERTology. What does the BERT model learn about natural language? A method of finding out if BERT learned something about another task B while being trained on a task A is called probing. We discussed it in a previous video linked in the description below. Until now, we have explained the BERTology part of the title. Where is the biology? Here it is. In the central dogma of biology, there are 3 important pieces that play a role in how cells live and reproduce: the DNA, the RNA, and the proteins. For the sake of this video, we can simplify this by saying that DNA is the blueprint of the cell, whereas proteins are the machinery built from this blueprint. The DNA contains a sequence of bases, often encoded as the letters A, T, G, and C. This sequence is then transcribed into RNA and then translated further into the language of amino acids. Amino acids are just molecules, but when connected into a long string, they form proteins. Similarly to how we encode bases as the letters A, T, G, and C, we can also encode the 20 amino acids with letters from the alphabet. This amino acid sequence is important for a well-functioning protein, but some amino acids have very similar chemical properties and could be exchanged without breaking the viability of the protein. Until now, we regarded proteins as a 2D sequence of amino acids. Only that the proteins exist in 3D space, how else? So important for the function of the protein is not only the amino acid sequence, but also the 3-dimensional shape that this long line of molecule takes. Biologists call this the secondary and tertiary structure. The tertiary structure is the overall shape of the protein. The secondary structure describes recurring subshapes such as turns and folds, helices, and sheets. Because proteins are folded into themselves in complicated 3D shapes, some amino acids are very close to each other in 3D space, even though they are far away from each other in the 2D sequence. And proteins don't all work by themselves, but sometimes they interact with each other and bind at well-defined binding sites. Equipped with this biological knowledge, let's look at how BERT could help us in protein biology. In natural language, we have text, which is a sequence of words. These words could form simple or more complicated meanings and patterns. For example, a simple pattern would be to identify that different words have a different part of speech. One is a verb, the other one a noun, and these patterns are not explicitly marked or annotated, but they contribute heavily to conveying meaning. More complex pattern would be coreference or sentiment, and so on. On the other side, we have protein sequences, where the words are the amino acids. Similarly, they also have intrinsic structural properties like the secondary structure, the tertiary structure, or the binding sites. So let us see if BERT, that works on language and identifies the inner structure of language with the attention mechanism so well, can also work on these sequences of amino acids and tells us more about the inner biological workings of the cell. The idea then would be to train BERT, like we train usually on language, by masked language modeling. We take the amino acid sequence, mask a portion of them, and then let BERT train while predicting what the masked amino acid might be. And this task would be for BERT quite simple. While English has a vocabulary of around 10,000 words, we have only about 20 amino acids to consider. Should be easy, right? Well, it's not that easy because we want to also learn something about interesting biology here. Reconstructing missing amino acids in the sequence is not our main goal. So for detecting interesting properties like secondary and tertiary structures or binding sites, the authors are analyzing the attention patterns. And they do this by measuring the correlation of the attention of each head to different properties of the amino acids. Like in natural language, some attention heads in BERT focus on only one specific word, for example on the or I in a sentence. For proteins, we have a head focusing on the occurrence of only one amino acid, for example proline. More interesting properties can be interpreted from this result. Attention heads that focus on one amino acid also have a high attention score for amino acids with which they can be substituted with. So amino acids with similar chemical properties share high attentions. This is similar to what we observe with synonyms in natural language. We can also find attention heads that align with the contact maps, meaning that in the three-dimensional structure, these amino acids are close to each other. So authors saw that this head's attention is a very well calibrated estimator for the probability of contact. For binding sites, this attention gets more complicated, since binding sites are more difficult to describe biologically, because there are many different types of binding sites. This is why there are more heads working together on describing this phenomenon. This paper is a nice proof of concept that many sequence-based data types can be processed by the transformer architecture. Without any explicit annotation, the model figures out interesting phenomena by itself. If you liked this video, please tell everybody about Miss Coffee Bean and do not forget to hit the subscribe and like buttons. Bye!